everyone, my name is Yen and I will be showing you the 3 different ways to transfer bacteria using the aseptic technique. So first things first, I made sure that I have all the proper PPE on. I have gloves, I have my lab coat, and I have my goggles. The very first thing that I want to do as I'm setting up is to turn on my microincinerator. It does take about 10 minutes to warm up, so I want to give it ample time before I need to sterilize anything. So I've turned it on, it's heating up currently, and while it's heating up, I'm going to sterilize my environment and make sure that it's really clean. I'm going to be using this alcohol spray on my surface here, and using a paper towel to wipe it down. I'm going to make sure that I have all my test materials ready. So I have my 2 petri dishes here. I'm going to make sure that I label them properly with the species of bacteria that I have in here. I'm going to make sure to write directly on the plate around the edges here, so that way I can still observe any bacterial growth in the middle. So I'll put my name, the date, and the species all around the corner here. I also make sure that I have my inoculating loop and my 2 test tubes with the caps on. And I want to make sure that the test tubes are not lying on their side. So I got this little handy dandy rack to make sure they're standing upright. So now that the microincinerator is warmed, I want to take my inoculating loop here and insert it right into the microincinerator. Hold it for about 10 seconds to make sure that it does have enough time to sterilize, and I'm going to make sure that the entire wire portion is inside the microincinerator. We just want to make sure that the entire wire is clean, since the tip of the inoculating loop is not going to be the only thing touching the bacteria or the test tube. And then after it's been 10 seconds, we're going to make sure that it cools down for about a minute or so. This allows the wire to cool down, and that way we don't have to worry about killing off the bacteria that we want to grab from the test tube or the petri dish. We're going to let that cool down for a second. Okay, so now that it's cooled down, I can now start getting ready to transfer bacteria. Okay, so I want to make sure that I grab the test tube, holding on to the glass portion. That way I have a very stable, steady hand and I don't drop the test tube or the inoculating loop. We don't want accidents, and I don't want to have to decontaminate everything again. So we are going to use the pinky to uncap this tube. A little tricky. Okay, we're going to set that down, and we want to make sure that the bacteria from the atmosphere and surrounding environment doesn't get into our culture. So I am going to hold the test tube lip or opening here just outside the microincinerator for 2 to 3 seconds to make sure that the opening of that test tube is sterilized. So once I have done that, I am going to insert the inoculating tube into the test tube, and we're just going to grab the bacteria, make sure that the loop gets in contact with the bacteria. Okay. And then once we grabbed our bacteria, we're going to sterilize the opening of the test tube again before we cap it. So we're going to hold it by the opening for 2 to 3 seconds. Cool. And then we're going to take this cap and cap it back on. Cool. So as we grab our test tube that we want to transfer this bacteria to, we're going to repeat that same method here. We're going to take off this cap. We're going to sterilize the opening of this tube, holding it for 2 to 3 seconds by the opening of the microincinerator. And then we're going to insert our loop into the test tube, making sure to do this motion. That way it allows the bacteria to fall off the loop and into this new test tube with new broth. Cool. And then we are going to sterilize the opening of the test tube again, holding it outside by the microincinerator for 2 to 3 seconds before we cap it. And we're going to set the cap. And then set it back into our test tube rack. And now we have successfully transferred bacteria culture from one test tube to another. And if we wanted to do that into a petri dish instead, we're going to sterilize our inoculating loop and insert it, making sure that the whole entirety of the wire here is inserted into the microincinerator for 10 seconds to make sure that everything is sterilized before we continue. Okay. We're going to open the test tube again. We're going to set it down here. And before we insert the inoculating loop again, we're going to sterilize the very top of this test tube, holding it outside of the microincinerator for 2 to 3 seconds to sterilize the opening of this test tube. This allows, the heat creates a convection, and that way any bacteria that could have fallen in gets pushed out. We don't want any bacteria from the surrounding atmosphere to contaminate our sample. Now that I have both of these things ready, we're going to grab some bacteria from this test tube. And before I cap this test tube, I am going to, again, set it right outside the microincinerator for 2 to 3 seconds to sterilize it before I cap it. Okay, I'm going to take the cap, close it. Great. And then I have my petri dish. So this petri dish, I want to make sure that I open it like a clam, like this, only halfway, to limit any bacteria from the atmosphere getting inward. And when I transfer the bacteria on my inoculating loop into the petri dish, I want to make sure that I am not holding it at a steep angle like this. If I do so, it'll just dig into the agar plate and it won't really be helpful, and that's not the proper technique. So I want to make sure that I hold it like so, and then I will streak across the plate hard enough so that it scrapes the surface of the agar plate, and then I'm going to close it. And once I close it, I'm going to put it into the incubator upside down. That way the water droplets that form will fall to the bottom and not contaminate or get into the agar gel that's in the plate. I'm going to do that. And then, of course, we're going to re-sterilize the inoculating loop by putting it back into the microincinerator for 2 to 3 seconds, making sure that the head and the handle of the wire of the inoculating loop is completely inside the microincinerator. Okay. After 10 seconds, we will let it cool down. And then for our last technique, we are going to transfer a solid material from a test tube into another test tube. So, same technique that we've used before. We're going to uncap this. We are going to make sure that our test tube is sterilized on the top. So we will hold it right outside the microincinerator for 2 to 3 seconds. Perfect. And if we had solid culture in here, you will see the material kind of at a slant here. And once our inoculating loop is ready and sterilized, and cooled down, of course, we don't want to skip that step, we're going to slowly insert the loop into the test tube and lightly scrape a small amount to be on the very edge of this loop here. We don't want to get a large amount of solid material onto the loop, just a small portion. And before I set the test tube down in the test tube rack, I'm going to again sterilize the top of it by holding it right outside the microincinerator for 2 to 3 seconds. Once that's done, I'm going to recap the tube, like so, and then place it back into the test tube rack. And if I wanted to transfer this solid bacterial amount onto a Petri dish, like before, I will make sure that this is open halfway, and I will make sure that I am not going in at a steep angle, and I will scrape across or streak across the agar plate and close it. Put it in the incubator upside down for the water droplets to fall and not contaminate or get into the agar gel of this plate. Once I do so, I am going to sterilize the inoculator loop for 10 seconds. Perfect. I will put this away where it needs to go in the lab, and I will be sure to use my alcohol spray again to disinfect the benchtop space I was just using. I want to make sure that this space is clean and ready to use by somebody else. And that is it. Thanks for watching.